Welcome to this video on bacterial multiplication. These are our learning objectives for this video. The first one is to be able to explain how bacteria multiply under optimum conditions, and the second is to be able to perform calculations involving numbers of bacteria. As always, all the timestamps will be in the description below, so you can skip to whatever part you need help with. There's no prior knowledge required for this video, but it is quite similar to the process of mitosis, so you might want to go through that to help you with this video. Moving forward from this topic, it also links really well onto culturing microorganisms, which is in your spec, but it's also a required practical. And in order to carry out that experiment, it's really good to have a thorough understanding of how bacteria actually replicate in the experiment, so you can understand why you're taking certain steps, for example. Binary fission is the process through which bacteria carry out cell division. It's similar to mitosis, but it's a means of reproduction rather than growth or repair of cells. So with a good supply of nutrients and the optimum temperature, bacteria multiply by binary fission every 20 minutes. They split into two every 20 minutes. So for example, say we start with one bacterium, and this is at time 0. So we have time here, and we have our number of bacteria here, so we have 1. After 20 minutes, we're going to have 2 bacteria as it's split into 2. After 40 minutes, we're going to have a total of 4 bacteria, as both of these original bacteria have split into 2, so we'll have 4. And then after 60 minutes, we're going to have 8 bacteria, as they have also all split into 4. So as you can see, binary fission is quite exponential in nature. The number of bacteria can increase very rapidly. So after a couple of hours, you'll have quite a large amount of bacteria. So if we are given the mean division time, for example, 20 minutes, we can actually calculate roughly how many bacteria will be present in a sample at the end of a given time using this formula, which is the number of bacteria at the beginning times 2 to the power of the number of divisions, and that will give us our total number for the n number of bacterias. And in order to calculate the number of divisions, we need to divide the time that the population is left for by the mean division time for that bacteria. So mean division time. So for example, if they're left for 3 hours and our mean division time is 20 minutes, so 3 hours is going to equal 180 minutes, so it's 180 over 20, so we're going to have 9 divisions. If, for example, we start off with 10 bacteria, so we'll pop that into the formula, so we'll do 10 times 2, and say we're leaving it for 3 hours, so we're going to have 9 divisions at a rate of a division every 20 minutes. So 10 times 2 to the power of 9. You pop that into your calculator, you'll get 5,120. So that is the total number of bacteria after 3 hours at a rate of 20 minutes per division. So you can see how big the n number is compared to the start number. That bacterial or binary fission is really exponential. So that allows the bacteria to reproduce quite a bit at in a really short period of time. Now we're going to go through the stages and processes involved in binary fission within bacterial cells in a little bit more detail. So starting at stage 1, we've got a typical bacterial cell with a cell membrane and a cell wall, and in the center we have our piece of circular DNA, as we don't have a nucleus because bacteria are prokaryotes, so they don't have any membrane-bound organelles. So our DNA is in orange and yellow, and the yellow part represents an origin of replication. So this is where DNA replication starts within the bacterial genome. So once we have that, in stage 2, our DNA is going to replicate and form 2 identical pieces of circular DNA. So for example, one of these will be our replicated DNA. As you can tell by the size, the cell has also started to elongate in preparation for division. So we end up with an elongated cell. In the third stage, we get more towards the division of the actual cell membrane and cell wall. So therefore, we have the formation of a septum in the middle, which kind of starts dividing the original cell into two halves, and the circular DNA kind of moves to each end. So we have the same amount of genetic material in each end of the cell. Then in the fourth stage, we finally have the division of the cell membrane and the cell wall, and this is pretty similar to the process of cytokinesis in mitosis. So then we end up with our identical daughter cells, which can then sort of go on to stage 1 again and repeat the process. And that is basically how bacteria replicate exponentially, because those two daughter cells that we produced can go through the same process, and then in total produce 4, and those will produce 8, and so on. In order to kind of see the process of binary fission in person and make calculations using bacteria, we need to be able to grow or culture our microorganisms in a lab setting so they can perform binary fission. As we mentioned previously, they only carry out this process if they have the right nutrients and the optimum temperature and generally the correct conditions. And we can use these two substances or kind of pieces of apparatus in order to do that within the lab. So the first one we have here is on the left is the nutrient broth. So this is essentially a liquid medium which has a suspension of bacteria in it, and then we also have a culture medium, and this basically contains lots of nutrients, as the name would suggest, which kind of encourage the growth of bacteria. So that will be things like carbohydrates, so for example glucose. We'll also have amino acids for protein synthesis, and then just kind of other minerals which are important in the growth and replication of bacteria. Then more commonly, when we're trying to see the growth of bacteria visually, or we're trying to kind of measure the effect of an antibiotic, for example, we use something called an agar plate. And this is essentially a clear, flat-bottomed plate, which also comes with a lid. And within that, we have agar jelly, which is a jelly-like substance which allows bacteria and other microorganisms to kind of grow on it. Itself is called a Petri dish, and then obviously we're going to add our agar jelly to that, and it's going to be, when we pour it in, it's going to be hot and it's going to be sterilized, so we have the no like previous contamination, so we're only going to grow the bacteria or the microorganisms that we want to see in our experiment. And in order to add our bacteria to this medium, we're going to use something called an inoculating loop, which is basically a kind of metal wire which we dip in our nutrient broth, and then we're going to kind of smear that on the agar plate in order to transfer our bacteria to our growth medium. These are some common mistakes that students make when they're attempting to answer questions about binary fission. So these are 2 top tips that you should keep in mind when you're studying this topic. Although it is quite similar to mitosis, a lot of these steps and the general function of the process is quite different. So number 1, binary fission is a means of asexual reproduction. So bacteria carry out this process in order to make more of themselves, which are kind of independent organisms. Whereas, for example, in humans, you might have cell division via mitosis for growth or repair. So it doesn't necessarily happen all the time. Whereas in bacteria, this is a constant process. It's a form of reproduction. Second one is that DNA replication and DNA separation happen at the same time in binary fission, whereas in mitosis and in part of the cell cycle, you have DNA replication in the S phase, and then you have the mitotic phase. In binary fission, those two processes occur at the same time, so it's not entirely the same. So these are our first worked example questions for binary fission. We're going to be carrying out some calculations using bacterial numbers after replication, as we went through before. So the first question is, a cell has a mean division time of 20 minutes. How many cells will it produce in an hour? So again, we're going to use the formula that we mentioned earlier. So that is the bacteria at start. So from the question, we can kind of assume that there's only one cell, as it says a cell, so we'll put 1 into our equation. The rest of the equation is then times 2 to the power of the number of divisions. So we'll do times 2. So the mean division time is 20 minutes, and we're leaving it for an hour. Therefore, 3 times 20 makes an hour, so we have 3 divisions. So our final kind of formula equation that we're putting in is 1 times 2 to the power of 3, which is 8. So after an hour, we're going to produce 8 cells. So the second question is a little bit different. It's kind of working out a different variable in our formula. So a single cell divides to produce 32 cells in 2.5 hours. What is the mean division time in minutes? So as we kind of mentioned previously, if we want to work out the number of divisions, we can do time over mean division time, which is put div time, and that equals the number of divisions. So in this question, we need to work out the mean division time. So we're basically just going to rearrange this formula and swap around these two values. So our mean division time is then going to equal our total time over our number of divisions. So as we know, our number of our time is going to be 2.5 hours as stated in the question. So we'll have 2.5 hours. If we write that in minutes, that's going to be 150 minutes. Then our number of divisions kind of needs a bit more working out. So it says that we've gone from 1 cell to 32 cells. So we need to work out how many divisions that is. And we can basically do that by doing 2 to the power of a number. That will give us our number of total cells. And just kind of trial and error inputting values into that, we work out that the x is 5. So that means we've done 5 divisions to get from 1 cell to 32 cells. So if we sort that back into our equation, we've got 150 over 5 divisions, and that's going to give us 30 minutes. So to answer the question, our mean division time in minutes is 30. This is our second worked example question for binary fission. So in this one, I'm going to ask you to pause the screen for a moment, work through it on your own, and then I will walk you through it. So a food manufacturer is growing a stock of bacteria to add to probiotic yogurt. To test their method, they inject a small sample of bacteria into a nutrient broth and monitor the number of living cells. Figure 2 shows how the number of living bacteria changes over time. At first, there is a lag period where the numbers do not change significantly. During this time, the bacteria are adapting themselves to new conditions. So when we first get a question with any sort of diagram or graph, it's important to have a look at it and just to kind of work out what it's trying to tell us. So on this graph, we have the number of bacteria on the y-axis and time on the x-axis. So we can work out that it's showing us how the number of bacteria is changing over a set period of time. So in the portion of the graph marked A, we can see that it's almost a straight vertical line. So we can tell that the number of bacteria is increasing very exponentially. In B, we've kind of got a horizontal line or a plateau of the line graph. So we can tell that over that period of time, the number of bacteria isn't really changing at all. Then on C, we can see that the number of bacteria is decreasing but it's at a kind of less fast rate than the increase in section A. So now moving on to the questions. Time point A takes place at what is known as the log phase. What is happening in the log phase? So as we've already kind of walked through, we can see that in the log phase, the number of bacteria is increasing exponentially. We've got an almost straight line. And we know that binary fission is also a process which increases the number of bacteria exponentially. So we can pretty much infer that what's occurring here is replication of bacteria, and that is going to be through the process of binary fission. Second part of the question is phase B is known as the stationary phase. Some cells are dying at this point, so why doesn't the overall number of living cells go down? So the question's told us that some cells are dying, but we also know from our answer in part A that cells are still replicating and reproducing. So therefore, if the overall number is not changing, we can deduce that the number of new cells being produced is roughly equivalent to the number of cells dying. So new cells kind of equal to old cells which are dying. The third part of the question is time point C takes place during the death phase. What is causing the bacteria to die? So as we talked through earlier, binary fission only occurs when bacteria have their optimum conditions. So that is having a lot of nutrients and also having the right temperature amongst other factors. So if cells are overwhelmingly dying and the number of bacteria is going down quite a lot, we can assume that these conditions aren't present. And if we look at how much replication has been going on previously, we can kind of deduce that it's most likely that there's not enough nutrients left for new cells to keep reproducing and proliferating. So we can say that there's not enough nutrients left in order for binary fission to occur. So to summarise, in this video we've been able to explain how bacteria multiply under the optimum conditions by binary fission, and we've also been able to work through calculations involving figuring out how many bacteria are present after a certain amount of time, or kind of how fast bacterial division occurs. We've also learned briefly kind of how to set up a bacterial culture so we can study how replication occurs. So this is a really good starting point for covering more information on culturing microorganisms and also for the required practical on this topic. So next, I'd recommend practicing some more calculations involving bacteria and then also going through that practical and the kind of next steps of the information.